Hello everyone. We are here to talk about HIV. What HIV stands for? HIV stands for Human Immunodeficiency Virus. Right. So, HIV is a retrovirus. What does it mean? A retrovirus is a virus that inserts a copy of its RNA genome into the DNA of a host cell that it invades, changing the genome of that cell. We will see how in when we will talk about the replicative cycle of HIV. First, we will see the structure of HIV. Let's draw HIV. So, I'm drawing it with a bright red color. Looks like sun though. And then we have these spike proteins. This is a typical HIV looks like. And this subunit of the spike protein is called GB120. 120. And this shaft part is called GP41. GP stands for glycoprotein. So that is glycoprotein 120 and glycoprotein 41 in the spike proteins. This one is nucleocapsid. Nucleocapsid. Inside of which there are two single-stranded sRNAs and then some proteins. We will talk about what proteins does it have, because it has three important proteins and then some other smaller proteins. So what these proteins are, let's see. The first protein is called reverse transcriptase. The second protein is called integrase. And the third protein is called protease. These are the three main proteins, and then there are some other smaller proteins too. So we rub this off and then we will see how each of these proteins function when we talk about how it replicates. So now we will start knowing about how it replicates step by step. So this is our red colored HIV with all of its spike proteins and the nucleocapsid and the genetic material, right? And then we have this big host cell, which can be a CD4 cell or a macrophage. This will also have its genetic material, and then there is a receptor on its membrane to receive the spike protein of HIV, and then there is one chemokine receptor along with other receptors. So this chemokine receptor can be CXCR4 or CCR5 depending on what type of immunological cell this is, whether it is a CD4 cell or a macrophage. So what happens? The receptor present on the membrane of this host cell recognizes the GP120 part of the spike protein. And then they will form a bond. Once this happens, once this happens, this GP120 part changes its structure, or it conforms, and it forms the structure like this, which extends and touches the chemokine receptor, makes a bond with the chemokine receptor. After this, what happens? Both the cells have come together, come very close, and now the the GP41 part helps in the fusion of the two membranes. When this fusion happens, what will happen? The genetic material, in fact, the whole nucleocapsid with the genetic material and the proteins move towards the host cell and enters the host cell. Once it has entered, the virus HIV leaves that space and it has already infected the host cell. This will leave now. Inside the host cell and inside the nucleocapsid, what happens that this reverse transcriptase enzyme helps in forming cDNAs or copies of these RNAs. So these RNAs, which are complement to the new cDNA strands, makes huge number of copies of them. And then there are this genetic material of host cell already present there. And we should have drawn this with red color because it is viral. After the reverse transcriptase has done its job, the cDNAs will move towards the nucleus of the host cell. Once it get inside, now that other protein that we talked about, integrase protein, will integrate the viral genome and the human genome. So this is this hybrid genome that you see, containing both the parts of virus and the human genes. Now this will produce a number of mRNAs and other RNA genomes. These mRNAs and viral genome are then broken down into proteins, viral proteins. This is done by the protein that we third protein that we talked about, which was protease. This is done by protease. So the viral protease and the human protease make some proteins. What are these proteins, by the way? These proteins are GAG, POL, ENV, and then other proteins. Inside the host cell, now all these proteins will assemble together, assemble to form nucleocapsid. With single-stranded RNAs and all of those viral proteins inside of it. And lastly, what will happen? This nucleocapsid will exit the host cell. And what this is called? These nucleocapsids are virions. Virions. So a huge number of virions are produced and it infects many cells of our body, causing AIDS. So I hope you got how HIV replicates inside our body. Thank you.